Welcome to your overview diagram of mitosis and meiosis. You should have already done the notes. So let's review first the difference between the cell cycle and mitosis. So the cell cycle is the entire life cycle of the cell, and it includes interphase, which is when the cell does its normal cell thing. So most of the time you'll see the cell in interphase, and then every once in a while it will divide and go through mitosis. So mitosis is the process of the cell dividing into two new cells. Now remember, mitosis is somatic cells, so these are your normal body cells. And some of your cells go through mitosis faster than others. Some divide like every 24 hours, and some divide, a couple times in your whole lifetime. So it just depends on the type of cell. So starting with interphase, we've already labeled that one. The cell's going to look normal. You can see all the organelles. Everything's doing what it's supposed to be doing. Toward the end of interphase, you get what's called S phase. And S phase is where the DNA replicates, and that's your signal that the cell is getting ready to divide, is when it goes into S phase. And you're going to learn all about DNA replication later, but the DNA replicates, meaning it makes an exact copy of itself, and then it will be ready to divide, but it still stays in interphase for a little while longer. All right, the first phase of mitosis is called prophase, and this is where the cell, the DNA will now form chromosomes, and at the beginning, while the DNA is in interphase, inside the nucleus, it just looks like kind of a bowl full of spaghetti, and that's called chromatin. But once it goes to prophase, it forms these little X-looking things, which are called chromosomes. So we're going to do two different colors here. Two of them are the original DNA, and two of them are the replicated DNA, but they're the same things, so that each blue one is an exact copy of the red one. Now, if we looked at a chromosome up close, let's draw that. Kind of looks like a little dragonfly. So there's some parts to the chromosome. So this is called a chromosome. Sorry, you have to kind of fit this out in the margins. Okay, so there's some different parts to this. So the middle thing is called the centromere. And then on each leg of the chromosomes, you got these little stripes, and each of those stripes represent a different gene. So these are the genes, and there's tons of these, thousands of them on each chromosome. And each leg of the chromosome is called a chromatid. Don't you love all this vocab? Okay, so in the box, we're just going to write chromosomes form. So that's all that happens in prophase, and pro means first, so prophase literally means first phase. All right, moving on to metaphase. Meta means middle. So metaphase literally means middle phase. And guess what happens to the chromosomes? They line up down the middle. Now, I forgot to mention that in prophase, the organelles kind of fade into the background and the nuclear envelope dissolves. And so all you can see are the chromosomes. All right, chromosomes line up down the middle. Now, in an animal cell, we've almost forgotten to mention our little friends, the centrioles. So those are those little churro things, remember? So the centrioles have these little lines attached to the middle of each chromosome. And so they use those to pull the chromosomes around during this process. But we don't have to draw them in every phase. Just know they're there and doing their thing. Okay, anaphase is the next phase. And in anaphase, the centrioles actually pull those chromosomes apart. So they kind of look like little parentheses now. So let's draw these little microfilaments. And here's chromosome number 1, and chromosome number 2, and number 3 and number 4. Okay. So chromosomes pull apart during anaphase, and they move to the sides of the cell. Now in telophase, the chromosome actually splits, or the cell actually splits. And this is called cytokinesis. So cytokinesis is not a separate phase, it's part of telophase. So first the chromosome kind of splits apart and it looks kind of like a little infinity symbol, and you've got the kind of half chromosomes going here, and then once you get back to a full complete cell, now all the organelles reappear and everything becomes normal again, except now you have a normal number of chromosomes. So when we started up here in interphase, we actually only had 4 chromosomes. You can't tell that because it's chromatin, but you just had 1, 2, 3, 4, and they were all, actually let me erase that because that is wrong. They should all be red. Okay, so 1, 2, 3, 4. And then they duplicate in S phase. And then we've got twice the amount of DNA here in prophase as you were supposed to have in a normal cell. So when we get to the end, we have 2 daughter cells. No, there's no such thing as son cells. Sorry, guys. And those daughter cells now have a normal amount of DNA. So it's the same amount of DNA that the original cell had. And that is mitosis. All right, here we are on meiosis, which looks a lot more intimidating, but actually will go a lot faster than meiosis or than mitosis because it's a very similar process. So meiosis is cell division of sex cells or gametes. And the idea with the division of sex cells or gametes is that gametes join together in sexual reproduction, and so you don't want a normal amount of DNA. You want half the amount of DNA that you would have in a normal cell because when they join together, you want to have a cell at the end that has a normal amount of DNA. You don't want something that has twice as much DNA as it's supposed to have, because that would mess up a whole bunch of things. So they have half the normal amount of DNA. All right, so normal cell is called a diploid cell, and a gamete is called a haploid cell. Haploid means half, and diploid, di, means two. So normal cell has a normal amount of DNA, and gamete has half. So, you know, if you wanted to make somebody think that you're insulting them, but you're not really, you could just say, oh, quit being such a diploid, because, you know, that's normal. So, all right, so let's start at the top. You had to deal with some Mrs. Brown jokes here. Okay, so if we start at the top, I've already got it labeled. That's interphase. Cell's doing its normal cell thing. This starts the exact same way that mitosis does. So we have S phase, where the DNA replicates, and it's doing its normal stuff. Okay, so here's the cell, and it's doing its normal stuff. Now, prophase, the DNA has replicated, so we have twice as much DNA as we're supposed to. And so here we go with 4 chromosomes, where the normal cell would have 2 chromosomes, and it forms chromosomes. So this is called Prophase One. We're not going to go ahead and label what happens in each of these because we already did that on the other side, and it's basically the same thing. Okay, so the next phase is called, you guessed it, Metaphase One. And remember, meta means middle. So here we go. There's a little bit of a difference, though. Whoops. Hold on. Instead of lining up in one single line, in meiosis, they line up in pairs. Okay, so that is a slight difference. Now, during this process between anaphase and metaphase, we have something called crossing over. So because they line up in a single line in mitosis, crossing over doesn't usually happen. But in this case, it happens quite often in meiosis. So what happens, let's draw a little close-up. Let's say we have a red one and a blue one next to each other. What can happen is sometimes the legs will cross over and touch each other, and a little part of them will actually break off. And so once they go into anaphase, you'll end up with what looks like this. You'll have a blue one with one red leg, and you'll have a red one with one blue leg. What this does is ends up making more variation in the offspring, because you've kind of mixed up the genes a little bit more. This isn't a mutation. This is a natural process that creates diversity. And so it makes it so that your offspring don't automatically get identical genes to their parents. So it's actually kind of cool, and that's why you don't always end up looking just like Uncle Henry when you grow up. So it would look like this. Make sure you're using colored pencils for this because you need these colors so that you can kind of see what's happening here. All right, so you'll notice they're still in 2 rows, and our centrioles are doing their centriole thing, and they're pulling things around. And this is fine for anaphase. And then we get to telophase 1, and there's still little X's. They aren't parentheses. Okay, and you have your red one and your blue one, and then your crossed over ones. And so make sure you keep them crossed over. Okay, so in telophase 1, now all of a sudden we're going to split off, but they're still X's. And so you'll have that over there, and then you'll have this one. So you see how now we've already got some diversity. All right, so telophase 1, and that ends phase 1 of meiosis. So now we're going to skip a step because we don't go back to S phase. The DNA does not replicate again. And this is how we're going to accomplish getting half the amount of DNA. So now we're going to go to prophase 2. And in prophase 2, the same thing happens as in prophase 1. The chromosomes form. But because the chromosomes are already formed, there's really not much to it. So this part goes pretty fast. So you got your 2 chromosomes, and there they are. Then we go to Metaphase 2. And here's where they do line up in a single line. Oh, and I grabbed the wrong color, sorry. Okay, so there they are. Okay, Metaphase 2, line up down the middle. Anaphase 2. They split. So now you're going to get your little parentheses again. But then we have this one weird one that's half blue, half red. Okay, and same with this side. You're going to get that and that. And you're going to get the little half one there. Okay. And then finally, we get telophase 2. And this one's going to have those. And this one's going to have those. This one's going to have those, and this one's going to have those. And now you have 4 gametes that came from one single cell to start with. And so you end up with 4 haploid cells that have half the amount of DNA. Now, if these are... All right, so at the end of telophase, you've got these 4 haploid gametes. Well, each gamete ends up being either a sperm or an egg, but each one is different. And so whichever ones end up joining together will have different traits. And so this ends up providing a lot of diversity for the offspring, and that's why you never know what you're going to get. And you're also at a 50% likelihood of getting a girl or a boy, depending on which chromosomes ended up with which gamete. So that's the end of this activity.